Let's see, those of you at home and here in the room, good morning. Let's talk for a minute about the exam. So those of you in the room have your exam. Those of you who are not in the room, please feel free to pick up your exam. Please remind me to give you your exam in lab this week so that you have it on Thursday or Friday. So many of you did really well. Congratulations to you. Many of you, maybe even those of you who did pretty well, feel like you didn't do as well as you were hoping for. And I guess I want to say a couple of things about that. One is, I do understand that this is challenging conceptual material. Conceptually, the material is challenging, and the detail is challenging. And we're doing it in half the time that it's usually done. So I do understand the challenges. But I feel that you're all up to it. I really do. I feel that I just want to remind you, for those of you who didn't do well, that there's many opportunities to bring your grade up. The quizzes, the next quizzes, the next problem sets, the next exam. So don't be discouraged. I do recognize that it's difficult material, and I want to encourage you to, ramp it up. Use if those of you who did well, whatever you're doing, do it again. Continue to do it. Those of you who didn't do as well as you wanted, practice learning with active strategies, rewriting notes, rewriting stories, sketching cartoons and labeling them. Writing and talking are active, reading and reviewing are passive. I'm a real strong advocate for doing active things. I'm happy to answer questions about the exam. The answer key is on Moodle, so you can look at your answers and look at my answers and see where I subtracted points from your answers. But I'm still happy to go over it with you, because some of these concepts will appear again in other assessments, whether it's on the final exam or in another section exam or maybe in a problem set. So I advocate using the exam as a learning tool and, adjusting if necessary, and not adjusting if you're doing things well. There is a problem set due on Sunday. The schedule says Friday, but that's too soon. So the next problem set, hard copy you might have in your hands, electronic copy on Moodle. It's a Moodle thing, so you download the problem set, you do it, and you turn it in on Moodle by 9 p.m. on Sunday. That's the next homework. Well, that's the next homework that actually has a turn-in, but I'm expecting you to be doing homework pretty much every day, even when there's not an assignment. Reviewing, rewriting, etc. So problem set due on Sunday, and then another quiz due on Tuesday. The quiz will be just like the last one. The quiz will be pick up at CST desk, sit down and do it, turn it in at CST desk by 9 p.m. on Tuesday. Okay, so things have got shifted back a little bit, but I'm going to get back on schedule for problem set and quiz 3. Okay, so use the problem set. Remember, problem sets are open book, open notes, open friends. Use the problem set to help reinforce the knowledge that I'm going to be talking about today. So today's material will be on the problem set, and you'll see you're going to use it. The quiz, the next quiz coming up, I want to say, will be on shoulder, arm, and forearm, and back muscles. So some review will be on the quiz. So don't forget everything you learned about the arms, because it will be on the quiz. So forearm, arm, shoulder, and back muscles. Your coloring book is your best source, and we'll reinforce that in lab this week. And brain. Somewhere in the schedule it might suggest that you would have, like a coloring assignment might be the I, because that's coming up on Monday, but I will be not on quiz 2. I will be on quiz 3. Okay? If you have questions about focusing your efforts, I'm happy to answer those kinds of questions too. Do I need to know this for quiz whatever? Do I need to know that for problem sets? Well, problem sets are open book. So, let's see. So you have another key concepts list. It's rather complete. It's a checklist of things that you should know. You have a hard copy of your problem set, but really the operational thing is on Moodle. Oh, and also on Moodle are your scores, your grades for problem set 1, quiz 1, and exam 1, but not for lab. So the total in the course is an underestimate of where you are right now because everybody's doing great in lab. And so the lab score brings everybody's score up either a little or a lot. Okay, so remember that the lab score will add to your total score right now, but it's not there yet. What's there is problem set 1, quiz 1, and exam 1. I don't know when mid-semester grades are due, but you have what you need. I'll do it because I'm supposed to, but you have what you need to know from Moodle. Okay, so let's get started. We're transitioning now to another section of the course, which is going to use the concepts from the first section of the course, but focus on sensory and motor systems. Those are the systems we're going to be focusing on for the next 2 weeks. And then there'll be another exam. So, this is physiology. So we think of sensory systems as gathering data, turning information out there in the form of vision, photons hitting our retina, or sensations on our skin, or sound waves hitting our ear, or temperature hitting our skin. Information input. We will talk a little bit about neural processing, but not a lot. And then we'll talk about motor output. So thinking about input and then output, motor systems. So it's a real simple view of a human being, because we're not talking about cognitive things very much, a little bit. If you were interested in cognitive things, take a psychology course, cognitive research, or there used to be a course in the psych department called Sensation and Perception, which builds on this, is less physiological and more psychological. So today we're going to do the somatosensory system. So we're transitioning to a system and we're focusing on one system today. The somatosensory system. The sensation of somato just means body, like soma is cell body, somato means is our body. The information from our body, touch, pressure, temperature, pain, proprioception, where our body is. We sense, I know my arm is extended even if I'm not looking at it. I can sense that it's extended. So we know we can sense our position. That's part of somatosensation. So we're going to start with some general principles and then move into the somatosensory systems. So this is basically, I'm not going to go through it. This is an outline of what we're going to do for the next 2 hours or so. Even though this is Wednesday, we're not going to do a breakout because it's the first class of the section. So sensory systems, general. We're going to talk about somatosensation today, then vision, hearing, smell, and taste. That's our roadmap. Here are some images of those systems. Here is a general concept that I want you to understand. Some of the sensory systems have a receptor, a sensory receptor, that is part of a neuron. And the neuron has an axon, and the axon sends action potentials to the brain, to the spinal cord or to the brain. Some of the sensory systems, the receptors are not on neurons per se. They're on sensory cells that release a neurotransmitter that tells a neuron information. So these are called long receptors because the cells are long, they have an axon. And these are called short receptor cells because they don't have an axon. So the division is that touch, well, somatosensation, touch, pressure, temperature, some proprioception, position sense, all of that are, all of those receptors are on long receptor cells. And the olfactory system, our sense of smell, all of when we detect chemicals in our nose, chemical odorants, those receptors are also on long receptor cells. The others, sight, hearing, tasting, the receptor cell is a short cell. Now, the mechanisms here are that receptor potentials trigger action potentials that take information to the brain. The concept here is that receptor potentials trigger neurotransmitter release, which then excite a neuron that take action potentials to the nervous system, to the central nervous system. So you can predict that, for example, these cells express voltage-gated sodium channels because they fire action potentials. They also express voltage-gated calcium channels because at their terminal, they release neurotransmitters. These cells express voltage-gated calcium channels because they release neurotransmitters, but don't express voltage-gated sodium channels because they don't fire action potentials. The receptor potential is coupled directly to this depolarization is close enough to this location so that this depolarization opens voltage-gated calcium channels and triggers neurotransmitter release without an action potential. And then excitation here, depolarization, threshold, voltage-gated sodium channels, action potential propagation. And then out here, in both of them, voltage-gated calcium channels, calcium influx, neurotransmitter release, the stuff you showed me on your exam. So here is a cartoon of a long receptor cell. So let's just say this is touch in my fingertip. A stimulus triggers receptors. We'll talk about the receptors for touch in a minute. Depolarization. Okay. So here's the stimulus, stimulus intensity, low intensity, medium intensity, high intensity stimulus. Low, medium, high intensity pressure stimulus, let's say. The receptor potentials, the depolarization of the receptive part of the cell is graded, just like synaptic potentials. So you should think of receptor potentials in sort of the same category as synaptic potentials and not in the same category as action potentials. Depolarization that tracks the amplitude of the depolarization tracks the magnitude of the stimulus. Then So this cartoon is a little false. This electrode should be here. And then here is the axon hillock, right, that determines whether this cell fires action potentials, but let's just pretend it's here. Okay, so a little bit confusing, but this is the receptor potential, and this is now whether or not it reaches threshold. So a small receptor potential, maybe not big enough to reach threshold, and this signal is going to die out as we move down the axon because we need action potentials to get all the way down here for any stimulus. So this is a sub-threshold stimulus that will not trigger any neurotransmitter release from the terminal of this cell. Here's a receptor potential that exceeded threshold, fired action potentials, caused action potentials to fire. Those action potentials propagate faithfully down the axon and trigger neurotransmitter release. Here, a receptor potential that is well above threshold, more action potential. So remember, the stimulus intensity is going to code for not the amplitude of action potentials, not the amplitude, but the frequency. So here's a higher frequency of action potentials propagating down the axon, triggering more neurotransmitter release because more voltage-gated calcium channels are open. So we code a stimulus first by a receptor potential amplitude, and then by action potential frequency, and then by the amount of neurotransmitter release, which is then going to trigger synaptic potentials in the next cell of the pathway. More neurotransmitter release, more effect on the next cell. Well, yes, question.
So at the synapse is it's like at the bottom right little graphic, when it's firing a lot of action potentials, will each one of those action potentials reaching the terminal correlate to an action potential in the following cell?
No, there's summation going on here. So these action potentials are going to get summated in depolarization here, triggering an amount of neurotransmitter here. There is going to be a correlation between the frequency of action potentials here and the depolarization, let's say, if it's an excitatory synapse, of the next cell. But there is not a one-to-one correlation between this action potential triggering an action potential in the next cell, or this one or this one. It's integrated at the synapse.
Cool, thank you.
Other questions? Another concept that I want to introduce is the concept of receptive field. And the idea that some receptive fields are small and some receptive fields are big. So this is a cartoon that is using the somatosensory system to illustrate some receptive fields. So if I stimulate, let's just say my fingertip. My fingertip, there's parts of my skin that is getting stimulated a lot. And then parts of my skin around the stimulus that are getting stimulated a little. So if I stimulate, and each of these sensory neurons is innervating a part of the skin. So if a stimulus is in the middle of the receptive field of this neuron is going to fire more action. It's going to get a larger stimulus, larger receptive potential, because more parts of the neuron are getting stimulated, more receptor potential, higher action potential frequency. In the margins, it's not zero. It's not there's sharpness, but it's not ultra sharp. There's overlap between this sensory neuron and this sensory neuron. And so a stimulus here is going to trigger a smaller frequency of action potentials in the neurons marginal to the stimulus. We have the same thing in the retina. We're going to have sharp visual fields and fuzzy visual fields. We're going to have sort of the same thing in the taste. We're going to have certain tastants that are very sharp and certain tastants that are fuzzy in terms of receptive field. I mean, I'm not talking about where on the tongue a tastant is triggering. I'm talking about your ability to say, that's lemon, versus, I don't know, is that lemon or is that orange or is that lime? You know, that's a receptive field issue in taste. Is this stimulus here or is it, you know, where is it? Is easy to do for somatosensory, but we have receptive fields for taste, for vision, for auditory. So the receptive fields, as you might imagine, in the sound are frequency. Some cells are really sharp on a certain frequency, and some cells respond more fuzzy to frequencies. Those are receptive fields in the different somatosensory, different sensory systems. So this is illustrating it for somatosensory, but we have it for all of the systems. Some neurons we're in the somatosensory system still, some neurons have big receptive fields, large splay of the receptive area, and some neurons have small receptive fields, sharp receptive fields. So for example, the sensory neurons that innervate the skin of the lips, mouth area of the face, fingertips, those have very small receptive fields. And so we can do very sharp two-point discrimination. Now, in the past, I like to bring students up here to demo this. We're not going to do that this year, but you can imagine, if I take this tweezer sort of thing, and I put the tips 1 millimeter apart, 1 millimeter apart, and I touch my lips, I'll be able to tell that that's two stimuli, because I can tell, I'm getting a stimulus from this neuron and from this neuron, and my brain says, oh, that's two different stimuli, that's two different points of contact. So I have a very small receptive field that allows me to discriminate two very closely positioned points that are stimulating the skin at the same time. But if I take this 1 millimeter and I put it on the back of my neck, or put it, the back of my back, I won't be able to tell whether it's 1 or 2. I'd have to separate them by about 1.5 inches, 2 inches, to be able to detect two stimuli, because the receptive fields on the skin of the back are much bigger. This was also fun to demonstrate, to show that they couldn't tell whether it was 1 or 2 when it was this far apart. I guarantee you that if we did it here, which we're not going to do, but we could do this in lab if you're interested, because we're shielded up. Okay, so we have small, sharp receptive fields and large, fuzzy receptive fields. Questions? Zoe.
So, if you have like a higher density of neurons in a certain area, does that mean that you're able to distinguish the 2 points with a higher precision?
Yeah, typically. If these neurons which can detect very sharp receptive fields, if for some reason these were all merged into one pathway downstream, then the answer would be no. But typically, when we've evolved an area with high receptive with high sharpness, with small receptive fields, so we can detect differences, these pathways stay segregated so that our brain can sense, oh, there's two inputs here. Whereas here, the same stimulus is only getting one input. Did that answer your question, though?
Yeah.
The next concept that I want to introduce, which is very important for most of the sensory systems, including somatosensory, but really important in visual that we're going to get to on Monday, is this concept of lateral inhibition. This is a key concept. So let's walk through this, because it is generally important for all sensory systems. Let's say there is a stimulus, sort of like here. There's a stimulus in the middle of the receptive field for neuron B. And it's in the margin for the neurons A and C. Okay, so this input is similar to this data right here. Clear? This concept of lateral inhibition is going to make it possible for the brain to sharpen receptive fields in the areas where it needs to be sharp, like on the lips or tongue, or fingertips, or vision, where we're focusing and we want to discriminate two very closely positioned points of light. So this is a mechanism. So one mechanism is to have receptive segregation. That's one mechanism. And the other mechanism is this idea of lateral inhibition. So we have a stimulus in the center of B that is triggering some action potentials in A and C. Okay? Now let's make sense of this stuff. First, let's think about action potentials in B. Let's look at the through pathway. So this is the recurring theme. I'm going to talk about the through pathway, and then I'm going to talk about the lateral pathways. The through pathway, action potentials, there's the symbol for a synapse, voltage-gated calcium channels, calcium influx, neurotransmitter release. It's an excitatory neurotransmitter for this at this synapse. So it excites the next neuron, and the next neuron faithfully integrates the information input. Okay, that's simple. And the same thing is true. We have the same pathway going on in A and C. But notice that B, the pathway B, the output, or the action potential frequency, the information coded in the second neuron in B, is relatively faithful to the stimulus. Whereas in A and C, the low stimulus, low action potential frequency, actually got lower because of lateral inhibition. And this means that we have sharpened the receptive field, because high action, the ratio of this to this is less than the ratio of this to this. If you count them up, you can do the arithmetic. This signal to noise is better than this signal to noise. How? Lateral inhibition. Let's go through that. This stimulus triggers action potentials in these collaterals. These collaterals activate interneurons. So here is a word that I will expect you to know. This neuron, the green neuron, expresses an excitatory neurotransmitter. Let's just say it's glutamate. It's probably glutamate. It's releasing glutamate here, which is exciting the secondary neuron. It's releasing glutamate here, which is exciting the secondary neuron. But the secondary neurons that are going lateral are inhibitory interneurons. These are GABAergic neurons. They're stimulated by glutamate, but they release GABA. Inhibitory interneurons. So when this signal propagates to here, and these action potentials propagate in the collaterals, it releases a lot of glutamate here, which causes a lot of action potentials here, which causes a lot of release of GABA here, which inhibits this synapse. And so neurotransmitter release here by this stimulus is inhibited by the inhibitory lateral pathway. And so less glutamate here, fewer action potentials in the secondary neuron. Same thing over here. Now you could say, well, wait a second. This stimulus is also inhibiting this neuron. True. But this stimulus is smaller than this. So this inhibition is smaller than this inhibition. That's why this is pink and this is red. This inhibition is smaller than this inhibition, sharpening. So better throughput on the main pathway and lesser throughput on the lateral marginal pathways. Laterally inhibited. Going from 4 to 1, going from 20 to 15. So some inhibition from 20 to 15 in this illustration. But 20 to 15 is less inhibition than going from 4 to 1. Lateral inhibition. Questions about this concept? We're going to see this in the retina big time, Monday. All right. Let's talk about neuroanatomy related to sensory systems in general. So there are parts of the brain that have functional areas that are dedicated to specific sensory input. So for example, in the very posterior occipital lobe is the primary visual cortex. There's going to be other parts of the brain that process visual information, but this is the primary visual cortex. And there's part of the brain in the superior temporal lobe, the superior temporal gyrus, that is the primary auditory cortex. There's going to be other parts of the brain that process sound, language, but this is the primary auditory cortex. This is the central sulcus. This is the post-central gyrus, which is in the anterior parietal lobe. So this is the front sulcus, front gyrus of the parietal lobe. The post-central gyrus is the primary somatosensory cortex. There's going to be a taste cortex and an olfactory cortex that are buried deep and really are not shown on this lateral view. So I do want you to know where the primary visual cortex is, where the primary, we'll talk about this later, where the primary somatosensory cortex is, and where the primary auditory cortex is. The name of this gyrus, or in this case, the part of the lobe that is important. Questions about the functional neuroanatomy that I want you to know. So now, let's take a 5-minute break, wake up a little bit, walk around, because now we're going to transition into the somatosensory system. I've been using somatosensory for examples for general concepts, but now we're gonna really transition right into the somatosensory system. So let's take a 5-minute break.